Hello students, welcome to EPG Pathshala. I am Dr. Shaukat Mir from Hamdard University, Delhi. Today we are going to talk about the module Quality Control of Herbal Medicines from the paper Novel Drug Delivery Systems Part 2. So students, let us see what we are going to learn from this module. So number one is the needs of quality control. So why we need this quality control? This question we will ask ourselves, and then we will try to look for the answers that we can get for what is the need of quality control and standardization of herbal drugs. In the same way, in the second part of this formulation, we will also try to understand what are the ways and methods of achieving this quality control and standardization. Under this, we will try to understand what are the guidelines, both national as well as international guidelines, that are acceptable in order to go for the quality control of these herbal drugs. In addition to these quality control guidelines, we will also try to understand guidelines with respect to the toxicological studies of herbal drugs. So, at the end, we will also try to discuss some of those hurdles, bottlenecks, or impediments in the quality control process of herbal drugs. So let's get started with the introduction of quality control. So let's first discuss what is quality. So whenever we talk about quality, actually we say like we want to know what is present and how much is present. So if I, in a brief way, if I try to understand what is quality, then I should say that it is a measure of content and purity of a substance. So this definition, it applies to any equipment, it applies to any substance. So when you want to know what is present and how much is present, you are actually talking about quality. And when you want to know about these by certain procedures, you are actually talking about quality control. So quality control means it is a set of procedures in order to ensure that the product that you have manufactured or the product that you have got, it adheres to the defined set of quality criteria. As we understand that for these medicinal products, there are three parameters of quality. They should be safe, they should be effective, as well as they should have uniformity of content. So a substance needs to be, a drug needs to be safe, it needs to be effective, it needs to be have a uniform content. So when you want to check these parameters in a substance or a drug, you are actually talking about quality control. So there is an associated term with quality control, what is called as quality assurance. So in quality control, you check the quality of the end product, while as in quality assurance, which is a related term to quality control, you include or you try to inculcate quality while the product is being manufactured. So we say that quality control and quality assurance ensue, they confirm, or they want to, these are the procedures to confirm that the product and procedures, they meet the quality requirements. One more term that I want to understand, I want to make you understand is standardization. So in standardization, you have certain procedures, and those procedures or frameworks, they are acceptable or they are discussed with all relevant parties, industrial setups, analytical scientists, and regulatory authorities. They all come to one point where they discuss and stick or adhere to certain limits of certain substances, of active substances, of toxic substances with respect to plant or drug quality. So this standardization thus represents a framework to which all relevant parties and stakeholders agree to adhere, so that all processes associated with the creation of a herbal product are performed within set guidelines. Let's now understand the concepts of quality control and quality assurance in more detail. As per WHO guidelines, herbal drugs must have efficacy, safety, and quality. However, the problem often associated with these drugs is the inconsistency of their content and purity. So, uniformity of efficacy, safety, and quality is paramount for worldwide acceptability of herbal drugs. So, to attain these, the herbal drugs need to be checked for quality, which is done by quality control or quality assurance. First, quality control. Let's understand this term. Quality control is a systematic evaluation of the extent to which a substance meets the specified requirements. It encompasses both product and process quality so that the finished product is efficacious, safe, and of reproducible quality. While as quality assurance includes set of procedures to ensure that the herbal product meets the specified requirements before the final or finished product is ready. Thus, we can say that quality control and quality assurance ensures control over product as well as process. This is a pictorial representation of quality control of herbal drugs. You see that several of those parameters, be it botanical, physical, chemical, biological, or organoleptic parameters, they are involved in the quality control of herbal drugs. So, botanical, as we understand, they mainly include the morphological as well as microscopical parameters, while as the physical parameters include moisture content, extractive values, ash values, fluorescence analysis, etc. Chemical parameters, they constitute both the qualitative and quantitative chemical examinations with respect to the contents as well as heavy metal pesticide residues besides mycotoxins and other toxin residues inside the plant product. The next set of parameters are biological, where we look for the microbial contamination, the toxicological residues, pharmacological efficacy of the drug, so as a quality parameter. Now let's go into the detailed discussion about the quality control parameters. As discussed earlier, quality control is a check on the content and purity. So there are checks at different points, like starting with the starting material, and there are certain procedures or tests that are done with the end product. There can also be checks and checks in the process that is in between. So let's first go to the material authentication parameters. So the inputs or the ingredients that are to be included in an herbal formulation or a drug, they are to be authenticated. So authentication is both, it has parameters related to its biological aspects. So what do you mean by biological aspects is that you want to know whether the substance or the material has been collected from a proper plant material, as well as whether the particular plant has been, whether the particular plant part has been collected. So this is known as material authentication parameters. So the exact plant, the exact part at an exact time. When you want to confirm these, we call it as plant material authentication. So botanical terms or zoological terms in case of animal products, they make material authentication more reliable. Number two is morphological and histological parameters. So after confirming the genuineness of the collected material, we now set it for some testing. So these tests, they can be both preliminary with respect to their shape, size, odor, or its color, which are organoleptic properties, or they can be little advanced, which are morphological or structural parameters, identifications, as well as microscope related parameters, so which are known as histological parameters. So the quality control of these drugs with respect to its structural parameters, whether or organoleptic parameters or microscopical characters, they come next in line. The third type of parameters that are used for the quality check of herbal medicines or drugs is physicochemical parameters. As the name suggests, that it is a combination of physical activities, physical characters, as well as chemical characters that are utilized in this evaluation. So physical properties like ash value, refractive index, uh, its melting point and boiling point. So when these are used for the analysis, for the estimation of quality, we say it as physical characters, physical parameters. While as the chemical components, the testing for chemical components or the chemical behavior of the components, so this constitutes physicochemical parameters of quality control. The fourth one is general quality parameters. So this is with regard to parameters related to the fineness of powder, for example, in case of powders. In the same way, for example, in case of tablets and in pills, it can be with respect to the disintegration or the absence of chipping of those corners. So uniformness of content, so they come under the general quality parameters. So pharmaceutical elegance of these herbal drugs, it is tested when we talk about the general quality parameters. And the last but not the least is determination of toxins. We understand, students, that these herbal drugs, they may have some inert substances, they may also have some of those toxins, toxic substances. So there are certain formulations, there are certain products for which there are limits of these toxins beyond which they should not be present. So when these toxins are determined in uh, products, we call it as determination of toxins. In the same way, these toxins in the form of pesticides, heavy metals, they also get included during the during the cultivation or collection of these drugs. So the determination of these pesticides, insecticides, heavy metal residues, or these other chemicals that are used during storage for sterilization. So determination of these toxic residues on the herbal medicines is determination of toxins with respect to quality control parameters. This is the quality control aspects of herbal medicine with respect to finished products. So when we have finished products, they can be subjected to pharmacognostical evaluation with respect to morphology, their botanical source or zoological source, as well as their biotechnological source. There are also parameters related to the phytochemistry as well as physicochemical parameters, besides residual analysis, which constitute heavy metals, pesticides, toxins, radioactive substances, fumigants, stimulants, etc. Now let's go into the details of WHO guidelines for quality control of herbal drugs. So they are guidelines which are accepted all over the world, because of which they have been selected, and they have been adopted by all major countries that follow quality control of herbal drugs. The first parameter is plant material authentication. So all herbal drugs need to be identified and authenticated by taxonomists, ethnobotanists, or traditional system practitioners. Voucher specimens should be deposited in the raw material museum or herbarium for future reference and referred by an unambiguous specimen number. Next follows botanical parameters. The first one is morphological characters. Related to botanical parameters, I must say that it is a systematic evaluation of drugs. So evaluation with respect to size, color, odor, taste and sound, on crushing, breaking or shaking, they constitute organoleptic properties, while as the structural description supplemented by diagrams, photographs, sketches are important part of the morphological description. Powder microscopy is another parameter with respect to botanical evaluation. Very small quantities of powder samples are observed under microscope after simple preparation steps, and the various parameters or various features are observed for authenticating the material that has been included in the herbal formulation. Sometimes samples are mounted with staining with some class specific reagents like use of iodine for the location of starch. Next is foreign matter evaluation. So herbal drugs, they usually contain foreign matter in the form of molds, insects, animal excreta, or even plant parts from the same which don't constitute the drug or from other plants. So usually the drugs should be free from foreign matter, but there are rarely certain samples that are free from foreign matter. So in order to look for foreign matter, we need to take the sample and pick the unwanted materials and then look for the percentage of foreign matter present. It should not be more than specified limits with respect to certain pharmacopoeia as mentioned in this pharmacopoeias. Next set of evaluations or parameters are physicochemical parameters. They include first extractive values. So this is the total yield of a substance to any solvent. So we can follow cold extractive method, hot extractive method, or successive extractive methods. So in cold extractive method, samples are macerated with different organic solvents in a closed flask for 24 hours with occasional shaking, and then solution is filtered and the filtrate is evaporated to yield a residue. So percentage of residue to initial material gives you cold extractive value, while as the hot extractive value is determined by extracting the sample in a Soxhlet apparatus with various solvents. The extract is evaporated to dryness and the weight of the final residue is compared to that of the initial material. While as the third one, successive extractive values, they are calculated after the sample is extracted with successively different solvents, and each time the extracts, they are evaporated and the residues, they are mentioned or compared to the initial dried plant material. Let's now discuss ash value as a quality control parameter. Ash that is left after incineration of the plant material in a silica crucible when the temperatures are about 400 degrees Celsius. They are usually carbon free and the total weight of the ash with respect to the initial material is known as total ash content, while as acid insoluble ash content is determined by treating the ash with dilute HCl for 5 minutes. The insoluble matter is filtered on ashless paper, washed with hot water and heated at a temperature below 450 degrees Celsius to get acid insoluble content. While water soluble ash value is determined when the total ash is dissolved in water and the insoluble part is collected by filtration on ashless paper, and the paper is heated at 450 degrees Celsius to a constant weight. The difference of the weight between insoluble part and the total ash gives water soluble value. Next is loss on drying. Air dried samples are dried on an evaporating dish at 105 degrees Celsius for 6 hours. The sample is weighed at intervals of 30 minutes. The drying is continued until two consecutive determinations match each other or do not show a weight difference of more than 0.01 grams. There are also certain content that are taken into consideration for quality control, like fat content, resin content, total phenolic content. So in all these determinations, the samples they are extracted, total phenolic content. So in all these determinations, the samples they are extracted or tested for fat, resin, or flavonoids or phenolics to determine the content of fat or respective substances. The last one is determination of pH value, where the sample solution or suspension of a 1% weight by volume or 10% weight by volume solutions are prepared and the pH is determined after filtering the extract with a standardized glass electrode. Let's move on to now journal quality parameters. As discussed earlier, these are parameters related to number one, fluorescence analysis. The fluorescence pattern of powder samples is studied after treatment with different reagents like sodium chloride, sodium hydroxide, hydrochloric acid, ferric chloride, etc. in both daylight as well as UV light. The powders are also studied after reaction with different reagents. The preliminary phytochemical screening is also carried out as the part of the quality control parameters. So where the samples they are extracted with solvents concentrated in vacuum and preliminary phytochemical investigation carried with for the detection of alkaloids, glycosides, tannins, phenolics, flavonoids, etc. Let's now discuss how to determine the toxic substances inside herbal preparations or products. First, there are toxins which are known as mycotoxins with respect to microbes. So first let us discuss the microbial load determination. In this, sample is suspended in sterile distilled water, shaken for some time, and it is filtered. After filtering, the filtrate is taken as stock solution and serial dilutions are prepared. The diluted solution is separately inoculated on an agar, nutrient agar medium and incubated at 37 degrees Celsius for 24 hours. After incubation, the colony forming units are counted by colony counter. So the limits for colony forming units in case of aerobic bacteria should not be more than 10 raised to power 5, and in case of yeast and mold should not be more than 10 raised to power 3. This pertains to heavy metal toxicity determination. As we understand that the heavy metals lead, cadmium, mercury and arsenic, they are very lethal, and if they remain in the herbal drugs, they will render it unfit for use. So the quality control procedures need to look for the content of lead, cadmium, mercury and arsenic. So under this lead, cadmium and mercury, they are determined by one method, while as the arsenic is determined by Geldal method separately. In the first determination related to lead, cadmium and mercury, the sample is cleaned is and taken into a clean silica crucible. Then it is digested with concentrated nitric acid and perchloric acid in a ratio of 1:1. The crucible is heated slowly, first to 100 degrees Celsius for 3 hours, then to 120 degrees Celsius for 2 hours, and very slowly to 450 degrees Celsius and maintained for 4 hours. Then the contents are filtered and the sample is subjected to atomic absorption spectroscopy for determination of these substances. In case of arsenic, the sample is digested with nitric acid and sulfuric acid, and nitric acid is again added drop by drop till no darkening of the solution takes place. The flask is allowed to cool. Distilled water is added along with ammonium oxalate. It is again heated and cooled. The contents, they are transferred to a volumetric flask and made up to a volume of 250 ml. The solution is filtered through membrane and the sample is subjected to atomic absorption spectroscopy for the determination of arsenic. Determination of aflatoxin residues is done by mixing the sample with methanol and distilled water. The contents are filtered and depigmented. It is then added with zinc acetate and aluminum chloride in distilled water. Then diatomaceous earth is added as filtering aid, followed by addition of sodium chloride and petroleum ether. The mixture separates into two layers. The lower aqueous layer is transferred into another separating flask and extracted twice with dichloromethane. The residue is analyzed for aflatoxins B1, B2, G1, G2 using TLC, mobile phase of chloroform acetone propanol 98.5 to 1:0.5 and detected under UV light for estimation of these aflatoxins. While as for determination of pesticides, recommendations of Food and Agricultural Organization and WHO, they are followed. So they vary from country to country, while as these pesticide residues, they are to be determined as the part of quality control exercise. Let's now understand toxicological studies. Toxicity studies are carried out for clinical trial for the formulation on at least 4 species. In phase 1 of clinical trial, acute and subacute toxicity studies are carried out for a few days only. For acute study, 4 animal species are taken. One should be at least the non-rodent group. While in subacute studies, 2 species are taken and test sample is tested at 3 dosage levels for 2 weeks. Biochemical parameters like lipid profile, blood sugar levels of test animals are monitored, besides serum concentration and pathological parameters. Phase 2 of clinical study is for 1 to 2 weeks, while as phase 3 studies for toxicological studies are carried out for 3 months to 1 year. Rest of the procedure is same as that of phase 1 clinical trials. Let's now understand the basic hurdles or bottlenecks of standardization of herbal drugs and formulations. The first and the foremost is that we understand that herbs are mostly multi-component formulations. Herbal drugs are mostly multi-component formulations, so this is the basic bottleneck which we face while standardizing herbal drugs. Next is that we need to establish the quality control parameters for each ingredient, which is a combination of several plant ingredients or compounds. So we need to establish the quality control parameters for both active as well as chemical markers in each ingredient. as well as in the final product. Number third is mostly the chemical and chromatographic patterns of components of ingredients overlap each other. So this is the other impediment or hurdle that is faced in the quality control of standard herbal drugs, that the chemical and chromatographic these patterns, they usually overlap. So they may give a false positive or false negative results. The fourth one is the stability of ingredients in the formulation is usually unknown. We don't know about the stability profile of ingredients or the ingredients in the at the start or at the in the finished product. Number 5, the variability of samples with respect to their quality is almost unavoidable. The ingredients that are included in these herbal formulations, they show as a routine the property of variation or variability. So one batch of ingredients, they don't match the other with respect to chemical composition. So this makes the job much more difficult. The last but not the least is that we don't have uniform manufacturing standard operating procedures for herbal formulations. Currently, very few SOPs have been developed and validated, and even fewer are followed. Every company or every setup wants to develop its own SOP without considering the general aspects of stability and efficacy. Let's also now try to understand the main causes of these bottlenecks. First one is improper botanical identification, non-coherent chemical types, collection of plants at undesired stage of maturity, poor post-harvesting processes, storage and impure components, especially the therapeutic components, and lack of validated analytical methods are the main causes of the hurdles that we face in standardization. So students, let's now summarize what we have learned about this module. Number first, that thing that we have learned is that quality is the basic parameter that cannot be compromised. So quality evaluation of herbal ingredients should be and must be done so as to make these herbal drugs acceptable worldwide. So at this point, the main concern about the herbal drug acceptance is that they don't follow quality. One batch goes good in treating a drug disease, while as the other batch may fail. So quality evaluation will actually inculcate the uniformity of action. So the quality evaluation of herbal ingredients as well as the final product should be done. Number 2 that we have understood of this module is that SOPs, which are standard operating procedures for manufacturing of herbal formulations, should be developed and followed for the preparation of these herbal drugs. So there are SOPs established for some of those herbal formulations and drugs, but for most of them the SOPs need to be developed, validated and followed. Number third is that the standardization is an important parameter for maintaining quality of herbal drugs. You cannot say that my drug is a quality product without following standardization parameters, without following quality control procedures. So you have to let your product being screened or examined for the quality for its better acceptance all over the world. In the same way, the fourth thing that we have understood is that the stability studies of these herbal products and shelf life determinations need to be done as the part of quality control procedures. Safety of herbal drugs should be established on the basis of toxicological and adverse effect reporting mechanism. So this means that we have to develop reporting mechanism. So this time we don't have sufficient reporting mechanism with respect to reporting the adverse effects of herbal drugs. So we need to devise uh, adverse reaction reporting methods for herbal drugs particularly. Besides developing effective protocols, scientific protocols for determination of toxicological parameters. And the last is we have also learned about that the efficacy with respect to pharmacological screening and clinical trials should be established and validated. Means the validated protocols of pharmacological screening and efficacy estimation, whether our drug is efficacious. It is not that on some local or some these parameters which are not scientifically established. It is to be established on a scientific scale in a scientifically validated manner. Thank you.